Yes, dear students, once again, let's proceed. Table Talks Education. Today, or this time, we are going to look at an example involving codominance. Example on, we call it a cross on codominance. Members, let's call it a cross on codominance. Cross on codominance. Cross on codominance. That's where we are. Then you write that when a snapdragon, when a snapdragon in brackets, and renum, when a snapdragon and renum with red flowers, when a snapdragon and renum with red flowers is crossed with one or with another snapdragon, with another snapdragon with white flowers, all plants with white flowers, all in F1 generation, bear pink flowers. When a snapdragon, also called andrinum, that is a species of a plant which bear flowers. When a snapdragon with red flowers is crossed with another snapdragon with white flowers, all plants in F1 generation bear pink flowers. Okay? I say what is happening. Then you say, show the results of F1 and F2 generations. Results of F1 and F2 generations. Okay? First of all, when we are representing genes that determine the characters which are codominant, what happens is that we use uppercases. In other words, we use uppercases of the letters or alphabets. We use capital letters because there is no allele which is recessive and which is dominant. So we said the alleles are codominant. So now here, in another case, we can decide also to choose a capital letter which will represent the color. And now here, what happens is that under such an instance, then the other different capital letters which represent the alleles that determine the characters will be written as superscripts on the other capital letter representing the color. So now I can say that, we say solution let capital C represent color. Let capital C represent color. Then let capital R represent the allele for red flowers. Allele for red flowers. And also let W capital W represent. So now, under such a circumstance where C is representing the color, then of course briefly here we shall write that let C carrying now R represent the allele for red flowers, and let C carrying W represent the allele for white flowers. So now I'm gonna have parents phenotypes. Here this is what I have. I have red flowers being crossed with white flowers. Then we can have parents genotypes. The parent with red flowers has genotype C. It has genotype CR and then CR. It is being crossed with C carrying W and C carrying W. So we are saying we can decide to choose one capital letter such as C to represent the color, and under such an instance, then the letters representing the alleles that determine the characters, of course, will be written as superscripts on the uppercase of the letter representing the color. And thus, from there, what will happen is that genes will segregate as shown here. So we shall have this, therefore, CR, right in brackets, and then CR, like that. Then also, we shall have another gene segregating and have CW and another C carrying gene W. So that is where we are. And of course, you have to see carefully on the board. Now at fertilization, what are we going to see? But we have gametes here. Gametes which are haploid, and of course meiosis is taking place here. And thus I can cross. I will make short cross lines as shown on the board. And also I cross this one with this, and also this one with this, as shown on the board. Thus we shall have CR, CW, CR, C carrying W, all over CR, then C carrying W. Also here we shall have C carrying R, and then C carrying, of course, letter W capital. Then lastly, we shall also have, of course, C carrying a capital R, and C carrying capital W. This is F1 genotypes. F1 genotypes where the diploid condition, which is 2n, has been restored. And now this happens during random fertilization. Random fertilization. Random fertilization. So, you can see that every offspring here is having a genotype RW, and those offspring bear pink flowers. And so I can write here F1 phenotypes. F1 phenotypes. Then I say all pink flowers. All pink flower. Now, of course, very fast, so let us proceed to F2 generation. F2 generation. Where F2 generation, where we have parents phenotypes. These ones here, they are pink flowers. We are crossing them with another parent with pink flowers. So, those are the phenotypes of the parents in the F2 generation. Of course, let us proceed. The genotypes of the parents, genotypes whereby they are diploid or diploid. We have we have C carrying gene R and C carrying gene white, gene W. Then also we have C carrying R and C carrying capital W, the gene for white. Now, the gene separates or the gene segregates during meiosis, as shown here. Of course, we form CR, we put it in a circle, and CW, also we put it in a circle. We are crossing it with CR, of course, after meiosis, after segregation, and then CW, that is after segregation of the gene. So now, what happens during fertilization? But each one of these are carried in single gametes, which are haploid. The separation occurs during meiosis, as shown on the board. Now, at fertilization, any one allele of the other gene can randomly fuse or combine with either allele of the other gene. And as we make cross lines, as shown here. So, if I make a cross line, crossing with this, and also the same gamete can fertilize the other one. So we come up to this one, here, where we shall make a cross line, as shown on the board. And also another one as shown. So it happens during fertilization, which is random, or can say random fusion. Random fusion, of course, of the gametes. F2 genotypes. F2 genotypes, where diploid condition has been restored. So what do we see here? We can see, of course, that this offspring will bear a genotype of CR and then C carrying R. C carrying R. Like that. Then also this one here, of course, it will bear C carrying R, and C carrying W. This one here, it will be C, genotype C, carrying, of course, carrying R, C carrying gene R, and another C carrying W. Then lastly is this offspring, whereby we shall have genotype C, W, and another C carrying W. That is the F2 phenotypic ratio. Now, F2 genotypic ratio. Now F2 phenotypes. F2 phenotypes, whereby we have one red, then we have two pink, and then lastly we have one white. Thus, finally, phenotypic ratio, F2 phenotypic ratio is 1 to 2 to 1. So, dear scholars, that is the F2 generation. Of course, we said that there is always a modification of 3 to 1 to 1 to 2 to 1.